Good evening. My name is Calissa Jackson, and I will be covering scientists modifying yeast cells and turning it into a cannabis tracker. So, over this presentation, we will be covering the introduction, which is going to be a summary of the article, the theories, which will form a connection between our textbook and the article, and our peer reviews, which is proving or supporting our main article. So, in the introduction, we'll talk about what is the purpose of our article and what the article is about. So, scientists at the University of Copenhagen Faculty of Science were able to modify yeast cells and were able to get those yeast cells to sense cannabis, and this could possibly now develop into a new drug test, which would be able to be done right through our iPhones. But how were they able to do this? So the scientists swapped out the GPCR in yeast cells and changed the genetic material. That way, the yeast turns red or glows when the yeast is exposed to cannabis. So, in part two of this presentation, we'll be going over the theories and how it connects to our article. So, in chapter 5, the research methods of biopsychology, this is also covered in our second theory. So, for theory number 1, gene knocking, excuse me. Gene knocking is a procedure created, a procedure, gene knocking, I'm so sorry, gene knocking are procedures creating organisms that have one or more additional gene. Through these procedures, we have opened new doors and new opportunities for research and therapies. Um, I feel like this is a good connection because it shows an example in the textbook from a 2008 study where Shen and colleagues used or created a transgenic mice which contained genetic material of a schizophrenic uh, family. The Scottish family had they showed major schizophrenia. And after swapping that DNA with the mice also began to show abnormal behavior. In theory two, green fluorescent protein, um, GFP is a protein that glows bright green when exposed to blue light. It was originally founded or isolated in 1962 after finding a jellyfish off of the west coast of North America and is now being used in biological research. It wasn't until the 1990s where Chefai and his colleagues used a transparent roundworm, replaced its DNA or replaced its genes with a GFP gene, and now the gene or the roundworm itself glows, and they use this as a marker for gene expression. And I felt like there was this connection between the roundworm and the yeast because the roundworm now glows green, and through genetic modification, they were able to make the yeast glow red to every time it senses cannabis. So now in part 3, we'll be covering our peer review articles. And unfortunately, I was only able to find 2 peer reviews. It was a tough time finding them, but I feel like these 2 reviews are very supportive of my topic. So in the first review, we have engineering G protein coupled receptors signaling in yeast for biotechnological and medical purposes. So in this article, it basically talks about how yeast with the GPCRs were now being able to use for pathogenic detection, drug discoveries, and can now be used for medical purposes. And with our article, we talk about how hopefully in the future with opioids, the yeast can now detect or can hopefully detect those. So opioids being like heroin, cocaine, hard drugs. And with this new research, it can help with things like drug stop during an airport or in an airport. So in our second article, the use of yeast biosensing, biosensing in the yeast was created or allows us to create fluorescent biosensors and use it for drug discoveries. Again, drug discoveries pops up when it comes to this yeast biosensing. It is so important and so influential towards our lives, and it can now be a new method for us when it comes to drug detection. So this is the end of my presentation, and here are my references. Thank you so much.